Hello. Yeah, we will be happy to record it. Sometimes people cannot come. Okay, my student maybe already or somebody started recording. So, is it okay with you?
Yeah, we actually want this.
Good. Okay. Hello. Hello, Bohdan.
Hello everyone. I can see only
Good.
myself, but it's fine.
Can you? Well, you can also see me. I suppose. Yeah.
Oh, hello.
Yeah. So. Yeah, this is. Do you have your presentation ready? You don't have to put it up yet, but then I will give the screen to you, and you will present with the share button, right?
Yeah, right. I have everything ready. Just to make sure you're ready.
Okay. So yeah, I would like to introduce you. So welcome everybody to another round of the bioinformatics seminar at the Brno Masaryk University, and we are a hybrid seminar, and we have some collaboration with a group from Ukraine. So you are the first speaker for this group. Maybe there will be one or two more. We'll see. So Bohdan is a graduate student in Kiev, and he finished his bachelor's degree in applied mathematics in the Kiev Polytechnic Institute, and he's currently a graduate student of bioinformatics and also carrying out research with Alina Frolova from the Institute of Molecular Biology and Genetics in Kiev, and also with Sergey Mangul in the University of Southern California. So welcome Bohdan, and the floor is yours.
Thank you so much, Matej. Delighted to be here. Now, today I want to talk about this exciting research that I was a part of. It's the topic of my research is called rigorous benchmarking of methods for SARS-CoV-2 lineage detection in wastewater. But I'd like to tell you more about the bigger picture of what the lab at which I'm at is working on, and what are the perspectives of the wastewater analysis. And so here we go. First of all, let's discuss two of the papers. They clearly show that this wastewater monitoring is a very interesting thing to talk about. Let's consider these graphs. So in this paper, scientists have been gathering wastewater throughout a period of 5 months. We can see here the green line represents the clinical samples, and like the amount of positive cases in clinical samples, and the blue line represents kind of the abundance of detected COVID variants in wastewater. And we can see good correlation here. So that's promising. Next, in the second research, we have it even more interesting. The picture is the following. We have blue dots, wastewater reported cases, and gray are the clinical. And then we see that first reported cases of alpha COVID variant was actually reported later than it was first reported in wastewater. And significantly so, we can say like a couple of weeks was the delay. And the same thing happened to the Delta variant. So we can, with confidence, say that this data analysis of wastewater can identify trends in disease spread and detect variants earlier. So that's great, but it has some issues. When we have low concentration of the of the specific variant in wastewater, we can have troubles detecting it, and also we can detect false positives in wastewater. So these are variants that are not actually present in real world. Let's consider another paper. Here we can see a graph that shows how the frequency of the virus in the wastewater sample relates to the predicted frequency of the virus when you analyze this sample. And they found that if we look at this graph, we can see kind of that the amount that there is in the sample, we can predict in using data analysis. We can see that also there's a logarithmical scale, so it's maybe not that smooth, but still pretty good results. Reassuring. And also, if the frequency, real frequency, is less than 1%, we have a lot of errors. So the detection limit is kind of 1% plus, but on real data it gets harder, 10%. As we can see, they studied both whole genome and spike-only data sets. These we also do in our research in the lab. Now I want to present you the basics of this kind of research. So what is strain, variant, and lineage? So if we have a variant, if we have a virus, we have a mutation in it, we get a variant. It's a change in RNA. Then if we have a change in virus behavior, we have a new strain. Simple enough, but let's consider an example. So we have a virus A. Two mutations took place. We got like variant A1 and variant A2. And variant A1 then later mutated into different ways, creating variant A11 and variant A12. So this A1 is a lineage having two ancestors, and as well as A2, if we consider it further. And then if A22, for example, is like researched to have to be, it is more contagious, like 50% more contagious, then we can call it a strain because behavior has changed of the virus. Then you might have seen the use of variant of concern, or VOC, on the graphs I showed before. So this is pretty straightforward. These are terms that scientists use to mark viruses, if for how interesting they are. So if variant of interest is something that might be dangerous, variant of concern is something that is certainly dangerous. And the criteria for variant of concern would be that we have clear signs, like epidemiologically. That means that we can see that the variant spreads very good. Then on the genomics level, the affinity of some proteins, of some spike proteins with some other proteins, it's a specific criteria, increases, then and it can be proven also in vitro. This is a criteria to market VOC. And VOI is just if it spreads well in certain communities, or it is detected in Europe, then we can call it VOI. Now let's talk about the pipeline that was present in the papers. First of all, they created a genome reference set. They gathered wastewater somehow, extracted RNA from it, sequenced it, and analyzed the data. The way they constructed the reference set, they used a GSAID database. It's the largest database in the world for virus, specifically called SARS-CoV-2 virus genomes. They took US origin genomes, 1,000 sequences per lineage, and excluded rare alleles, which makes the task a bit easier for them, I guess, of detecting them later. Then to collect wastewater, they used these. Well, basically they're cleaning the sample and doing all this wet lab stuff with that. Then extracting RNA from it. I won't get into details of the wet lab. Then they use Illumina sequencing. I'm sure you are familiar with it. I saw previous talks were considering it. But yeah, in short, we the fragmentation of genomes, then specific molecules called adapters are introduced to the fragments, creating this adapter ligated fragments. Then those fragments are got stick to the plates, which do so that they multiply, and then we use different fluorophores to then start getting signals from each individual fluorophore group, and this way sequencer kind of understands what fragments, what sequences are present. Sorry for this drawing. I used the resources I could for it, and here we basically, this is my pipeline, but also there is a future plan, and the future plan is the plan that has been done in the papers I've talked. Actually, it's using real DNA, using sequencing, but in my case I used synthetic data sets, which I've created using a specific tool called SimSec, which basically generates reads, which then we use to analyze and benchmark my tools. So, all right, we have a reference genome. Then we have some parameters we need to specify for good creation of this data set, synthetic data set. We put it in SimSec. It makes us, with some additional steps, a mixture of reads. That is a mixture which contains reads from different genomes from the reference. Then we use haplotyping tools. In my case, I was tasked to benchmark these three, but there are more. Then we get found variants. We then evaluate our results. So, yeah, we evaluate, and also all right, that's right. Yes. So one of my mentors, Sergei Knyazev, wrote a bunch of papers actually leading to what we now do in this lab. Sergei Knyazev, he actually was co-creator of one of the methods, ClickSNV, which is used to detect these samples, these variants in the mixture by using some complicated mathematical methods, statistical methods. And he actually did a similar work when they considered HIV genomes. HIV is 9,750 base pairs long. That is 3 times shorter than SARS-CoV-2 genome, which is about 30,000 base pairs. So the task was a little bit easier, and they benchmarked the same tools, and they get some I guess interesting results. The precision is not terrible. Even they got 1 with perfect precision. on one of the data sets. I hope you can see my cursor. And
Yes.
oh, great. Yeah, this ClickSNV has a lot of top scores according to this table from his paper. Yeah, and these data sets are also mixtures of HIV variants, synthetic mixtures. Let's now talk about my pipeline. It's a little bit broken, the text. 1 second. Okay, so I have to do a lot of setting up at first, because every tool requires a specific approach to it. I'll talk about it later. Then we generate reads using Simsack and Python. We align reads, creating a map of reads on the reference genome sequence, like so. We can see small reads here, and they are mapped onto a sequence on top. Then we run the tools and collect outputs, align variants with the reference. Yeah, we then need to all right, we'll talk about it later. For this, there is a number of modules, a good amount. For example, Bioconda was very handy, as well as some tools, which is part of it. I used BWA Aligner, which is a classic. Picard to operate with FASTA files and validate them, and Anaconda was essential for my work. I also want to introduce you to pipeline management systems and tell you what does Snake make. Actually, in my proposal for the internship, I had to use SnakeMake as a Python tool, Python-based tool, because the internship, the organizers really wanted me to use Python because it's a modern language, and in my proposal there was just a bunch of bash scripts in the outputs, and yeah, and I included SnakeMake. So what is it? It allows for creation of these rules. Let's for example consider a case where we want to align fastest to the reference. Sorry, align FASTQ reads to far reference. We can create such rule which would specify input files, output files, and shell command, which creates the output file. Like this, we can create then these kind of scripts where input, when rule O, I believe, is indicates the start rule, and if we have two samples, A and B, our pipeline, when we run a single command of SnakeMake, will look like this, where I guess they wanted to we want to after alignment sort the BAM file, and then index BAM file, which are useful operations to increase some certain tools' efficiency on our data. Yeah, so SnakeMake executes these two pipelines in one command. And we even can, if we add another even step to it, if we already have an index created, the SnakeMake will consider that file with index and skip the step completely, going straight to the fourth step. Simulating reads. I already mentioned SimSec. It's a pretty good tool and a used one. I would say well kept up to date. I just the parameters were 50k reads. I think we can benefit from setting this amount to a much larger number in the future, like 500k reads. We will see. And Illumina mode. It can run in different modes. It has a lot of parameters to think about. And this is my mentor's repository, which I used in this one. This is the one where HIV mixtures were used to benchmark ClickSNV and two other tools I showed you before. It came very handy for me, some of the scripts there. Then what I did, I selected 5, I believe, variants belonging to 5 different lineages: Alpha, Beta, Delta, Omicron, and Lambda. Genomes, of course, from GISAID. 1 sequence per lineage, yeah. Here they are. We use them to create these data sets I created. We have full genome and spike region dataset. We can see that spike region is much smaller, which makes it easier to operate with this mixture of reads with on the cluster. Much less reads. Sorry. Is it less reads? No, it's probably not less reads. But yeah, the amount of base pairs matters. I created 2 datasets of full length and a number of them of spike region. As you can see, currently my repository is 50% Python and a little less shell scripting, like Bash. The scripts I created were like calculating distance between sequences, like Hamming distance, Earth-Mover distance, which is a more general variant of Hamming distance. Generating reads script, which I actually borrowed from Sergey, my mentor, and scripts for cutting and aligning sequences, and other helper scripts, like setting up the environment and running tools. And the first iteration of my results looked like the following. We had some variants detected. like we have 3 tools here on top and 2 mixtures, spike mixture and full genome mixture. We can see that the tools, one tool detected 1 variant, the other 7 and 5, and the third 2 and 3. And in total there were 5 variants in each of the mixture. So it so happened that none of the tools detected, like, reset. This Earth mover's distance was high for every variant detected when we map it, when we compare it with variant genome. So these variants detected are not, doesn't look like the variants from a reference. We can see some huge distances here. And the second iteration, I used multiple sequence alignment to align these reads, no, these reference, these FASTAs. Yeah, so it improved results dramatically. Using multiple sequence alignment, we accounted for deletions and insertions in the genome and made it so that everything starts counting from one certain position on the general reference of SARS-CoV-2. So like in this case, from here, it would start counting distance, because here we don't even have a sequence. So it's not very good to consider this. Yeah, we still got 0 results, actually, when we talk about precision and recall for every tool. But the distance became much smaller. Yeah, there is still space for improvement. Yeah, so in the future, we can benchmark a lot more tools like that, but a good thing would be to tune the parameters a bit and get to the to good distances first. These are the tools from my mentor's GAIN paper. He just found all these tools to collect them to see what is out there, and we see a lot. And we can use pipelining to like snake-make to fasten up the workflow and make the results reproducible, and also we would be able to easily expand the our pipeline if we want to add another tool. Let's say we would have a lot less headache to add that if we had something like snake-make into play. The challenges I faced was that this was a new area for me, genomics. I come from applied mathematics background. I studied for my master's like second-year masters in bioinformatics, and yeah, not a lot of biology knowledge was in that applied mathematics bachelor's. And then I want to talk about poorly supported software. Some tools have very little docs and little discussions on the web, which is unusual to me, as I'm used to actually working with machine learning and using popular frameworks, maybe web development, and there is a lot of info on that, but with these methods it's not much. Also, the software required is a little bit old. One requirement was GCC 4.5.2, which is 2010, and I needed to install Python 2 for one of the two. And the combination of these two is also coming into play. If you have little docs and all dependencies, you get a fun time. Remote position, for me personally, it's a bit of a challenge. Less communication, less opportunities to discover something new in a new area for me is a con. And I wanted to mention, you were curious, Matej, what is the situation for science in Ukraine right now? And we have something like the following. We have ruined infrastructure. We've seen cases of ruined universities, even not in the eastern regions. Then currently we have some electricity problems. It's working on a scheduled basis. So every house is scheduled to have some downtimes. It's about 8 hours a day, but frequently it's more like 4 hours a day or less even. It depends on your luck. And then poor internet connection, it comes hand in hand. And when the downtime is in play, mobile connection also struggles, because maybe everyone is switching to mobile, or some connections spread their devices, some antennas turn off. I don't really know for sure. Then logistics is complicated. If you're doing wet lab, you can't buy reagents that easily. We don't have planes flying in Ukraine, so everything is delivered on trains or on automobiles. Lack of budget. We have a lot of money coming to fix the damages that the war has rained on us, and a lot of it goes on the army to protect our lands and minimize the damages in the future. Yeah, and some institutions have sent people for unpaid vacations, like in our lab in Kiev. This happened. Yeah, and people are stressed. That is more personal, but yeah, it's true. When you're reading these news about your hometown being under attack, it's stressful for for sure. But despite of that, I think people are staying strong. Some of them are even more motivated to bring to keep the science of Ukraine alive, to move it further even than it was. Like my mentor Alina, she just traveling in Europe right now, seeking collaborations with people, attending conferences. So yeah, it's not over. Yeah, I think that's it.
Okay, thank you, Bogdan, for the nice presentation.
Thank you.
Yeah, we think, at least I think of you almost every day, like what's going on with the war. So maybe I will use that slide and under your talk put some links because otherwise it would stay hidden inside the presentation. So we have some guests that were listening. So are there any questions for Bogdan? There's also a question and answer button if you want to write it down, but there's not. Okay, I had two as I was listening. So one part you showed this Click SNV tool and some other tools how they were benchmarked, and I was wondering, like, what kind of errors are happening that from which the benchmark suffers. So is it that the reads don't get assembled, or that they have too many errors? Have you any idea, like why in some percentage of predictions there are errors?
Yeah, we have some ideas. We will test them. For example,
Yeah, maybe
we have this feature of SIMSAC that, as you can see on the edges here of the alignment, we have little coverage, little sequencing depth. So we can actually work on improving it by just adding some appendix to this reference, and yeah, it will just cover this region much better than maybe there are some minor mistakes or mistakes in our methods. We'll be looking into it closely, and yeah, we're still kind of researching.
Yeah, thank you.
You're welcome.
I think I can add to that because, as Bogdan mentioned, it might be too little of data for the methods to accurately detect variants, because 50,000 reads, it's for all 5 variants. So it's about 10,000 reads per variant. Like in theory, it should be enough, but maybe methods were developed for different type of data initially. That's why it's nice when you benchmark to try to generate different types of data sets synthetically. I mean, when you're simulating, because Well, usually when people create methods, they report that their method is the best. And it's not always intentional. Sometimes you are biased because it's your software, and usually you show the best results on best suitable conditions for your method. So we will see whether they perform better if we increase simulated data size. So yeah.
Okay. Yeah. Thank you for the comment. By the way, I know I have some contacts to a group in Czech Republic, which is like an unofficial group of scientists who put together a weekly or bi-weekly report on the strains and the lineages that are present in the Czech Republic. So they are called the COG. But anyway, we can get into touch later. But they probably have, some people who already encountered some of these problems that maybe you only will encounter later.
Interesting. Can you link it to me?
Yeah, actually, I think we don't have too many. Maybe people can think about asking another question.
Yeah, sure.
I will look at the address. Okay, now I remembered. No. Okay, I will have to send it to you later because now it skipped my mind totally. Sorry.
Yeah, sure.
Yeah, I just wanted to say also a general remark that currently it's kind of harder to get funding to sequence COVID patients regularly because some governments think that we no longer need it because we are past like the critical stage in this pandemic. That's why some kind of massive profiling like we can do with wastewaters is really the methods we actually need right now. However, as you can see, bioinformatically, it is challenging, especially if we talk about de novo variants detection. Because if we know what to find, what to search for, it's one thing. But if we don't know, sometimes it's hard because it's not a perfect sample, because now here we basically simulate data and we have like a clean experiment. However, when you sequence wastewater, you're not getting like full genomes or nice data. You will be even more like restricted with the data type you get. That's why it's important to benchmark and maybe propose better tools now before it's too late, because you never know. Maybe like in half a year we will get new very aggressive variant. We don't know, so it's better to be prepared.
Sure. So yeah, so it's a nice way to circumvent the legislative problems or whatever of getting some information from the environment. Okay, so one more chance to ask a question, and let's wait a little bit. Okay, so thank you, Bohdan, for the nice lecture, and
Thank you.
we have one more week of guests in this seminar. Then there will be a break for our students, and then hopefully we will come back. So just watch the website as usual. Thank you to everybody for coming. Thank you, Bohdan, and see you next week.
See you.
Bye-bye.
Bye.